The research introduces the THEA-Code, an autoencoder-based IDS-correcting code for DNA storage. It leverages an end-to-end autoencoder for integrated encoding and decoding, addressing challenges through techniques like the differentiable IDS channel, entropy constraint on the codeword, and auxiliary reconstruction of the source sequence. This approach marks the first instance of a deep learning-based code independent of conventional coding frameworks in the IDS-correcting domain. Comprehensive experiments, including an ablation study, affirm the effectiveness of the THEA-Code. The research introduces a novel end-to-end autoencoder solution for an IDS-correcting code, addressing complexities in error-correcting studies. It builds upon techniques such as a differentiable IDS channel, entropy constraint on the codeword, and auxiliary reconstruction of the source sequence. This approach converges and reports commendable results, marking the first end-to-end neural network-based IDS-correcting code without reliance on conventional codes. The framework is depicted in Figure 1, showcasing the encoding of source sequences into codewords, IDS channel errors, and decoder reconstruction. This work diverges from traditional methods by employing a transformer-based model for the IDS channel and an entropy constraint on the codeword, offering a new perspective in the field of DNA storage and error correction. The proposed THEA-Code framework consists of a deep learning-based encoder, a differentiable IDS channel, and a deep decoder. The encoder takes the source sequence as input and generates a codeword. A random error profile is then generated, recording the positions and types of errors on the codeword. The IDS channel modifies the codeword into a corrupted codeword according to the error profile. The deep decoder decodes the corrupted codeword back into the sync sequence. The optimization target is the cross-entropy loss between the source sequence and the sync sequence. To address challenges, 3 techniques are proposed: a differentiable IDS channel, an entropy constraint on the codeword, and auxiliary reconstruction of the source sequence. The differentiable IDS channel is implemented using a sequence-to-sequence model, trained independently before the encoder and decoder. The IDS operations are promoted onto the unit L1 sphere in the L1 space to align with probability vectors. The differentiable IDS channel plays a pivotal role in optimizing the model IDS, theta. It generates SIDS and SIDS, which are then utilized to train the model using the Kullback-Leibler divergence. The significance of this channel lies in its ability to be optimized independently, making it a crucial component of the overall system. To address the challenges of quantization and information loss, an entropy constraint is introduced on the codeword. This constraint is based on Shannon entropy and is integrated into the optimization target during training. This approach ensures that the model is trained to minimize the loss of information while optimizing the channel. The optimization process involves finding the optimal parameters theta that minimize the Kullback-Leibler divergence between the true distribution and the model's output. This process is facilitated by the differentiable nature of the IDS channel, allowing for efficient gradient-based optimization methods. In summary, the differentiable IDS channel and its optimization are critical components of the model, enabling efficient training and minimizing information loss through entropy constraints. The paper addresses the chicken and egg dilemma in joint training by introducing an auxiliary reconstruction task for the encoder. This task involves generating a longer sequence that is then split into a codeword representation and a reconstruction of the input source sequence. The auxiliary reconstruction loss, defined as the cross-entropy loss between the reconstruction and the input sequence, is integrated into the overall loss during training. The encoder and decoder are implemented using transformer-based sequence-to-sequence models. The training phase consists of 2 parts. First, training the differentiable IDS channel, and then training the remaining components of the autoencoder with a weighted sum of different losses. In the testing phase, the differentiable IDS channel is replaced with the conventional one, and performance is evaluated based on metrics measuring disparities between the generated and actual sequences. This novel approach mitigates the chicken and egg dilemma, allowing for more effective joint training of the autoencoder components. The paper presents the performance of a novel method in generating DNA sequences for storage, leveraging a 100-long 4-array sequence as the source sequence. This approach yields an impressive 1.6 × 1060 candidate sequences. Due to the scarcity of closely related works, the focus is on ablation studies to evaluate the method's efficacy. Experiments are conducted with a codeword length of 150, aligning with established DNA storage practices. The nucleobase error rate, NER, serves as a key metric for performance evaluation. Given the large sample set, training, validation, and testing data are not explicitly separated. The source code will be publicly available, ensuring transparency and reproducibility. The paper reports the best results among 5 runs for each setting, highlighting the performance against coder rate for different source lengths. Additional insights into the validation NER across training epochs are provided through figures and tables, demonstrating commendable performance for certain source lengths. This research contributes significantly to the field of DNA storage, offering a promising approach for efficient sequence generation. The authors investigate the effect of varying coder rates on the performance of the autoencoder-based DNA storage system. By keeping the codeword length fixed, they observe that lower coder rates relieve the burden on the deep model, leading to better overall performance. Experimental results, presented in Table 1, show a trend of increasing NER from 0.36% to 6.07% as the coder rate increases from 0.47 to 0.93. The authors also conduct experiments with coder rates above 1, finding that the autoencoder maintains its performance, trading off IDS errors in the codeword for substitution errors. When the source length is 160, the NER is intuitively expected to be around 6.25%, with an additional 6.70% introduced by IDS errors in the codeword. The results suggest that by applying an outer conventional error-correcting code to rectify nucleobase errors, a practical IDS-correcting code can be achieved, comparable to existing heuristic IDS-correcting methods in DNA storage. Figure 3 illustrates the NER against training epochs for different source sequence lengths, showing that as the coder rate increases, the models are more likely to converge to a local minimal point, resulting in less competitive performance across different runs. The authors also explore the optimization of the hyperparameter lambda, which dictates the weight of the entropy constraint in the overall loss. The results of experiments with different choices of lambda are presented in Figure 4, showing the entropy constraint len, reconstruction loss LCE, and validation NER for each run. The optimization of the model's parameters to achieve low entropy and good reconstruction loss is a crucial challenge. Careful tuning of the entropy constraint weight is essential to avoid local minimum convergence. Experiments have shown that a weight of 0.01 is optimal, allowing for good convergence, low entropy, and maintained decoder performance. The page also discusses the effects of the auxiliary reconstruction loss, evaluating various combinations of identity mapping, difference, and positive sequence concatenation on validation NER across different training epochs. The authors of the research paper propose a novel approach to training error-correcting codes, suggesting that the encoder and decoder operate based on distinct logics, with each component's training relying on the maturity of its counterpart. To initialize the training process, an auxiliary reconstruction of the source sequence is introduced in the output of the encoder, imparting a basic logical ability. This auxiliary reconstruction involves a hyperparameter micro, which represents the weight of the auxiliary reconstruction loss locks in the overall optimization objective. The reconstruction R is independent of the mainstream of the autoencoder, having indirect relations with the codeword C, as both are generated by querying the same transformer model. The embeddings corresponding to R and C interact when applying the attention mechanism of the transformer. Experiments were conducted with varying choices of the hyperparameter micro, including micro equals 0, absence of the auxiliary reconstruction loss, and micro element of 0.5, 1, 1.5, different weights for the auxiliary loss. The results, as shown in Figure 5, indicate that without the auxiliary loss, all training runs fail, producing random output. However, when the auxiliary loss is introduced, the models converge well, and the evaluation NERs exhibit a similar convergence to a value of about 1%. This suggests that the application of the auxiliary loss is essential, but the weight for the loss has little influence on the final performance. The authors also explore the idea of imparting the encoder with higher initial logical ability through a more complicated task beyond sequence reconstruction. They adopt commonly used operations from existing IDS-correcting codes, such as forward difference diffs and position information encoded sequence POS, and attempt to recover the sequence from these operations using the encoder. The results, as shown in Figure 6, indicate that the reconstruction of the identity mapping I outperforms diff and POS, and the combination I plus POS exhibits the best performance. The authors draw connections to previous works on DNA-based information storage, citing Church et al. 2012, Goldman et al. 2013, Grass et al. 2015, Ehrlich and Zielinski, 2017, and Organic et al. 2018. DNA data storage has emerged as a promising technology, but it's fraught with error correction challenges. This paper provides a comprehensive review of various methods employed in this field, including the use of artificial chromosomes for data storage and high-scale random access on DNA storage systems. Forward error correction techniques are crucial in DNA data storage, particularly in correcting single indel or edit errors. Linear time encoders and order optimality play a significant role in this regard. The authors discuss several error correction codes, such as hedges, which can correct indels and accommodate sequence constraints. They also explore the application of autoencoders, unsupervised learning, and deep architectures in DNA data storage. Furthermore, the paper examines the use of deep learning-based channel codes for point-to-point communication channels, including the turbo autoencoder. Attention mechanisms in neural machine translation are also investigated. In addition, the paper delves into Entropicon's trained residual vector quantization and various error correction codes capable of correcting deletions, insertions, and reversals. The authors highlight the importance of correcting single indel or edit errors in DNA-based data storage, emphasizing the need for efficient error correction techniques. This comprehensive review underscores the challenges and opportunities in DNA data storage and error correction, providing valuable insights into the current state of the art in this rapidly evolving field. In the realm of communication systems, error correction plays a crucial role in ensuring reliable data transmission. Recent research has explored various coding techniques to mitigate errors in DNA storage and wireless communication. Polar codes, for instance, have been employed to facilitate efficient error correction in DNA storage systems. These codes leverage the concept of channel polarization to construct reliable channels from unreliable ones, thereby enabling accurate data retrieval. Segmented edit error correcting codes have also been developed to tackle errors in DNA storage. By segmenting data into smaller blocks and applying edit operations, these codes can correct errors and ensure data integrity. Furthermore, DNA-EON, a novel coding scheme, has been proposed to address constraint adherence and error correction in DNA storage. This scheme utilizes a combination of coding techniques to ensure reliable data storage and retrieval. In wireless communication, bit loss and gain correction codes have been designed to combat errors arising from signal degradation. Synchronization strings have also been employed to facilitate efficient data transmission. Moreover, researchers have explored the application of neural networks in digital communications, demonstrating their potential in error correction and detection. Machine learning models have also been investigated for their ability to generalize across different wireless communication domains. This domain generalization enables models to adapt to diverse communication environments, enhancing their robustness and reliability. Additionally, deep learning techniques have been applied to decode linear codes, showcasing their potential in error correction and detection. Overall, these research papers highlight the significance of error correction in communication systems and demonstrate the efficacy of various coding techniques and machine learning models in mitigating errors and ensuring reliable data transmission.